The cell cycle is the process in which cells go through to grow, replicate their DNA, and divide into two identical daughter cells. It consists of two major phases, interphase and mitotic phase, which is also known as M phase. The first part is interphase, which is split into three different categories. The first is G1 phase, where the cell grows and performs its normal functions while preparing for DNA replication. The second is S phase, where DNA is replicated, creating two identical copies of each chromosome. The third is G2 phase, where the cell grows further, checks for DNA errors, and prepares the machinery needed for division. And interphase takes up most of the cell cycle. This other phase is mitosis, which is part of M phase, and it is where the division of the nucleus takes place and has four main stages. The first is prophase. This is where chromosomes condense and become visible under a microscope. The nuclear envelope then breaks down, and spindle fibers form from the centrosomes. The second is metaphase, where chromosomes line up against the center of the cell, which is called the metaphase plate. Spindle fibers attach to the centromeres of each chromosome. The third is anaphase, where sister chromatids are pulled apart to opposite poles of the cell by the spindle fibers, and each chromatid becomes an individual chromosome. And the fourth is telophase. The fourth and final is telophase, which is where the nuclear envelope reforms around two sets of chromosomes. And this is where chromosomes begin to uncoil and the spindle fibers disassemble. After mitosis, the cytoplasm divides in a process called cytokinesis, which results in two separate cells. In animal cells, a cleavage furrow forms, pinching the cell into two. In plant cells, a cell plate forms, eventually becoming the new cell wall. The last thing I will talk about is the mitotic index, which measures the percentage of cells in a sample undergoing mitosis. It is calculated using this formula. Mitotic index is equal to the number of cells in mitosis divided by the number of total cells times 100. This is an important tool in research, particularly for studying cancer, as rapidly dividing cells have a higher mitotic index. Thank you for watching my video, and I hope you enjoyed learning about mitosis and the cell cycle in general.